All right. I think we will go ahead and get started. So I am very pleased to welcome our invited speaker for the Motivation Cognition meetings for today. Our speaker is Dr. Steve Chang, who is an associate professor of psychology and neuroscience at Yale University. Dr. Chang's research program aims to understand the neural circuit mechanisms of social cognition and social decision making using naturalistic social interaction paradigms as well as cutting-edge behavioral and neural technologies. Dr. Chang received his undergraduate and graduate degrees at Washington University in St. Louis and completed a postdoc at Duke University before beginning his faculty position at Yale. He's published groundbreaking research in journals including Nature Neuroscience, Neuron, and Journal of Neuroscience. And he's been recognized twice with the Innovation Award from the Social and Affective Neuroscience Society. He's also served as past president of the Society for Social Neuroscience and has mirrored other contributions. And so we're very pleased to welcome Dr. Chang today to the Motivated Cognition meetings and look forward to your talk. Thank you so much.
Thank you, Kimberly, for a nice introduction. Yeah, so happy to be here. Today I'll talk about some kind of new work from the lab looking at cooperation in marmosets, which are pretty interesting. So let's get started. So in our lab, we've been starting to think a lot about primate evolution in general, because I think more you study social cognition, it's really hard not to think about why certain species are that way. And I think social structure that's a product of evolution is very, very important for that role. So in our lab, we focus on two species. First one is rhesus macaques. And they are, very despotic species with low social tolerance, but they're also very social, but in different ways than marmosets that I'll talk about today. Marmosets are New World monkeys, and they are very well known for kind of altruistic behavior, prosocial behavior, and they're cooperative breeders. So they're kind of very different than macaques, and they're very family oriented. So I'll focus on that species today. And in particular, I'll tell you about their behavior being diverse and flexible in terms of how they cooperate with one another. So I think that's a very important thing for us. They're not just doing a specific task, but they're really flexibly adopting the strategies depending on whom they're working with. So they're trying to work together in this situation. So why marmosets? This is actually from Zurich, University of Zurich. Judith Burkert and colleagues show this very nice plot that I really like. If you plot the extent of allomaternal care, so this is like a care for young outside of mother, basically, and plot that with proactive prosociality. This is proactively being prosocial, like helping and caring and so on. You see that we're pretty good over here, right? But macaques and chimps are very competitive species. They're not that great in this kind of two-dimensional way of looking at things. Marmosets, on the other hand, are pretty good, getting closer to human in this two domain. So it's a great, interesting species to look at kind of prosocial interactions. So who are marmosets? They are New World primates, so they live in Brazilian Amazonian forest, and they're cooperative breeders. What that means is they kind of help raise young together in a family group cooperatively. And they're very socially tolerant. They're not that aversive to losing competition, for example. And they're really well known for prosociality. Because they're small, it also allows us to study more naturalistic behaviors. We can have them run around and interact and so on. And also housing is easier because they're small. And for neural recording side, they have this smooth cortex, so they don't have this fault, so it's much easier to target certain areas and layers if you are interested in that information flow. So here, like one of the first thing we did was trying to solve a problem that it'll be great to do really naturalistic experiment, but sometimes traditional setup that are used in that kind of setting is not kind of optimized for neuroscientific investigation. So for example, I'll introduced the cooperative pooling paradigm, which is really well known, marmoset pairs really don't do that many trials, or any animals actually don't do that many trials. And also, when humans come in and put food on the tray and so on, it's distracting, and it can be disruptive to their behaviors. So this is a traditional corporate pooling paradigm that I was talking about. It's well known in evolutionary comparative biology, psychology, anthropology, because it's been just worked very well characterized for a long time. And importantly, all the species known to cooperate, even species that are cooperative hunting, they all do this. So wolves do this, hyenas can do this, birds and elephants, and of course, humans and chimps. So here's a video of them pulling this, and you can kind of appreciate that, it's a heavy tray they have to pull together to get the reward. So they have to work together. One animal just cannot do it by themselves. Um, and then what's interesting is this is not just a kind of fixed pattern type of behavior, because when one animal is not hungry or not motivated, the other partner will try to encourage them. You can kind of see here. So he put hands on to go, and the other animal is still not interested. Turn around, and then okay, come on, let's do it. So they start doing it again. So there's definitely this flavor of let's work together to get this done kind of information there. So the problem is they don't do that many trials. So what we designed was to bring this to neuroscience kind of framework so we can actually get many trials. So we developed this apparatus called Marmoset Apparatus for Automated Pulling, Marmo app. And basically we automatize the pulling behavior by making this servo motor-based lever where we can control specific forces and acceleration and everything to the system so we can optimize their pooling behavior so that they can do many trials. And in fact, like they prefer to pool, they are tree living species, so they grab and pull the tree branches all the time. So we apply that. And this is like the setup where the boxes can sit and animals can be here. And on the screen, we could display information such as inequities, reward size, or things like that if we are interested in that direction. And here's like how they might look in a setup. So you can kind of see that they're, pulling levers and trying to engage with one another. I'll go more into detail of how these behaviors look, but the first thing is that with this setup, we can get a lot more trials out. So this red dotted line is basically mean number of trials per session from non-automated pulling tasks in the literature. So they do maybe like 10 and 10 per day. And that's not that enough for neural recording or behavioral characterization. But this one, we can go up to like 150 pulls for self-reward, and cooperation, we can combine that to more than 100 trials. So how do we train these animals here? So basically, you put two animals into the setup, and then they first have to learn how to, get the juice. So here, they just have to pull the lever, and the contingency between the two animals pulling is totally independent. So there is no correlation needed. And they just pull, and they get little marshmallow juice mix, which they really like. So they learn to pull for juice. Once they can do this, we go to the training. We call it mutual cooperation training. And we have three phases. First is L3. That just means two animals need to pull within 3 seconds of one another to get the reward. And then once they hit 50% successful rate, which means out of all the pulls, 50% of the pull that animal did was actually cooperative pull. Then they kind of graduate to the 2 second version and to the 1.5 second version, all the way to the 1 second version. We stopped at 1 second because the time required was just enough for them to coordinate and pull without making it too difficult or without making it too easy. And then once they get to this point, we call that mutual cooperation is trained. Okay, so I'll show you what they actually look like when they're doing this task. And here you can kind of pay attention to, not their lever, they're going to reach and pull. and the juice will be coming from here and going to this basin. But also look at them, how they are like paying attention to each other. I think that becomes pretty important later on. Successful. They're looking at one another. They're really fast. They're so they use head gaze, not eye gaze per se, right? So they're looking at one another, getting successful. So unlike humans or macaques, their orienting gaze, social situation or else, is all dependent on head gaze. And the eyes are used to stabilize the fixation after head gaze. The head that becomes very good for us because head gaze is a lot easier to measure and a lot easier to quantify in this kind of naturalistic setting. So how do they do this, right? So if you look at the success rate here across these training conditions colored to L3 to L1, you can kind of see they start going up, and then we go to the 50%, go to the next stage, they drop a little, go back again, and then next stage they drop, and then they kind of go back again, and then final stage they drop and go back again. You can kind of see if they can maintain this success rate. Um, and then interval is an interesting one. This is just the timing between the two animals. And I'm including all the data here, and you can kind of see interval is decreasing as they should because they're learning this trial to be closer to one another. And the reward intake actually goes up, even though task is more difficult, meaning that they're learning the task and they're doing the task so that, self-reward is always highest because there's no contingency at all between the two animals. But across this learning stage, you get a better reward count. We wanted to make sure like they can do this, right? But we wanted to make sure what kind of information or strategy they're using. So we tested two types of controls here. One, we call it no vision control. This is to test how important the vision is between the two animals or even looking at the lever. Could they completely rhythmically do this without any visual information or not? That's why we wanted to test here, no vision. And then we tested this automated lever control, which is kind of interesting one because we basically block the access of the lever of one animal, and this lever is moving automatically based on this same animal's previous schedule, a previous block or something like that. The reason why we wanted to test this is, twofold. First is, can they just look at the lever and react and match it? That's a very fast kind of cue. Another reason is, are they just using some kind of variable schedule pulling? So we wanted to rule out that kind of, non-socially derived way to solve the task. And these are all trained animals. So they start with mutual cooperation, no vision, mutual cooperation, automated lever, and mutual cooperation. And now I'm going to show you the video of these two conditions. And if you can kind of like, compare that to how good they were during mutual cooperation. So here's the no vision control. You can see the block is there. They cannot see each other. They cannot see the lever either. And in this condition, these well-trained animals feel very kind of lost. Like literally, like they don't think they don't understand what's happening. It seems like, right? So they're really sometimes they get it, of course, because coincidental success. But their behavior seems very off. They're trying to look, but there's no one there. For example, they sometimes get it. And this is an automated lever control. So this part is blocked, but this is moving based on his own previous schedule. And here, one thing to keep in mind is that where success rate is basically how many pulls you do that is successful. So when this moves a lot by itself, or this moves with him by itself a lot without successful, that will lower the success rate. So you can kind of see that in this condition. Sometimes they get it because they got lucky there. But this is keep moving, which is lowering the success rate. So how do they do this in this behavior? It's pretty clear that they need vision and they need social vision, it seems, because they cannot do this well based on just the vision of the lever. So here's the data. You start with high success rate, and then you drop when you go to no vision and kind of stay low, and then you recover when you go back to mutual cooperation. And going back to automated lever, you drop greatly and then stay really low for up to like 10 some days we tested, 11 days or something. And this is an interesting point to make because, if we are doing it right, we'll figure it out. And okay, I'm gonna look at the lever now, and I'm just gonna like time myself to the automated lever. But that is just not intuitive to these animals because that's not how they solve the task. They require the other animals' movement as a holistic way to actually get, information to build and reach some kind of decision, which I'll come back later. And then you go right back when you go back to mutual cooperation. So you can see that success rate is dramatically different across three conditions. Also, interpool interval is increasing as gets animals are in these two conditions, and reward count is also low from as expected from this. Given this, we can now starting to examine what kind of strategies are they using. They're doing this well, but they might be using some strategies. So we started looking at this from two different angles. One is from the pooling perspective, another is the social gaze perspective. So if you look at the interpool pool-to-pool correlation, this is cross correlation plot, you can see that on the automated lever, there's like absolutely no correlation. Um, no vision, there's some, but much weaker. Whereas mutual cooperation, there's a nice cross correlation. And you can kind of see p correlation effects here. The mutual correlation is highest, the other is low. And then if you plot the density of the correlation, you can see that mutual cooperation is the one that has a nice peak. And there's like second peak here. That's because, there's a leader follower dynamic. So if one animals tend to be leader, they tend to have a earlier peak there. And this is interesting time to mention that, who is the leader between dominant and subordinate animal? It turns out that subordinate animal tend to be the leader, dominant tend to follow. The reason is dominant is more motivated and they want to make it work together with a subordinate. Subordinate is like sometimes the bottleneck. So dominant being willing to, I'm going to follow you to make this work is one way to make things work between two pairs. It's like, when you teach your son or daughter how to play soccer first time or they're not good at it, you just don't want to play soccer like you're a professional. You want to actually follow them to actually play some soccer. That's the idea. Another thing we notice is that there's some kind of time signature of pooling. So if you look at this plot, this is one animal code named Kanga, and you see that autocorrelation of pooling behavior is really high in the beginning by design, but then there's some kind of peak 5 to 6 second-ish time course that peaks autocorrelation. And in fact, all the other marmoset pairs we looked at, there have some similar peaking around 5 to 6 second time scale. So that's interesting. It means that, within the 5 6 second time scale, there's some kind of rhythm there of pulling. And you can see that that's definitely something real compared to shuffle, as you can see here. So to us, this suggests that, at least if you just look at the pulling behavior, there's some kind of rhythmic pulling behavior is there at least. We can think of this as more like a pulling rhythm strategy. And I'll come back to this strategy after talking about the gaze. So we can also track animals' gaze. We can make this 7 markerless tracker on the face. This is combining deep left cut and antipose. And you can basically put a orthogonal line from the plane to know what they're looking at because they use social gaze. And now when you do this, all of a sudden you have two interesting time series. So we have this animal one's pulling time series, animal two's pulling time series, but now yours have animal one's social gaze time series, animal two's social gaze time series. So we can now try to link these time series to think about how they are working together. So if you look at the social gaze rate over learning, it increases over learning. So that's a sign that as they're solving the task, they're actually starting to look at the other animal more. And that's actually the really low for the self-reward condition. Another way to look at if this gaze behavior is kind of meaningful is you can look at this condition that I showed you earlier, automated lever control, where the information is all in the lever, right? Not in the animal anymore. So we can also look at how much they look at the lever. So this plot is showing partner's lever gaze over social gaze. So higher the value here means they're looking at the gaze more than the partner. And you can kind of see the partner automated lever condition. They look at partner's automated lever a lot more than mutual cooperation or self-reward or the self automated lever itself when they are not doing the task. So gaze behavior seems to be strategic. Even though they look at the right lever, they still don't perform well though in this automated lever condition. Um, to look at the social gaze dependent strategies, we can start to look at how they relate to success rate. So here, I'm plotting the mutual cooperation trials and plotting success rate as a function of social gaze count. And you can see they're kind of negatively correlated, which means that if animals doing really well, they actually look at each other a lot less. So one way to interpret this is like if you're doing really well, there's no reason to keep monitoring others to keep doing the task. So we can interpret this as almost like a reduced reliance or reduced vigilance when success rate is high, and maybe pull-in rhythm strategy is more useful in this kind of context. And you can quantify this better looking at the time periods of when the social gaze is happening a lot and when lever pull is happening a lot. You can see that compared to success pools, failed pools have a higher correlation between these two time windows, consistent with the idea that when you're doing well, lever pulling is happening a lot, you tend to look at the other less. Another way to think about social gaze is how precise it is. So that's a different way of aligning the data. So we can align temporally using cross correlation again, how tightly coupled to the specific action of the partner to social gaze, which is a little bit different than previous one, which is looking at kind of overall success rate. But this is like pool by pool alignment. And when you do that, you can see that compared to self-reward control, mutual cooperation have this cross-correlation effect here, and stronger time locking for partners pools on mutual cooperation, which makes sense. What's more interesting here is that when you align the data in this kind of pool by pool level, they have a better alignment with social gaze and partner successful pool compared to failed pool here. So this suggests that when you look at the precision of timing, you have a much better precision when partners pulling successfully. So that's an interesting point that when you're registering partners pulling successfully, so I'm going to also follow and pull the lever, for example. So if you put these two and two together, there seems to be two types of social monitoring going on. We call one of them kind of vigilance mode. That's when successful rate is low. So you need to have really like look at each other to communicate and let's get these things back on track kind of level of behavior. But also this temporal precision time locking, we call this partner coupling mode, where you need to track partner successful pools to keep doing pools successfully. And these are happening, at different time scales. But it's interesting that there's two types of social gaze use in this task. So we talked about three strategies here, how to put all this together. So basically, we took a computational modeling approach. We used a dynamic Bayesian model, DBN, and this is a graph-based model that can detect causal dependencies of different graphical nodes. So let's start with how we do this. So now we have pool time series for each animal, social gaze time series of each animal, and then we basically put this into a dynamic Bayesian network model for them to keep going in iteration to discover temporal causal dependencies between these nodes. And if it survives multiple iterations, then it becomes a stable directional edge, for example. And this is also MTW just means multi-time weighted. So basically, you can do this at 1 second time period or more or 2 or 3 seconds. So you can discover these causal dependencies at multiple time scales. And now whatever's emerged, right, from this DBN, that's one way to think about that is a strategy. That is a quantified view of the strategy that these animals are using. And with those, you can actually do subtraction of different conditions to find this delta value of this graph, let's say between mutual cooperation and self-reward. Then you can kind of starting to see that these edges, these causal dependencies are specific to mutual cooperation, but not to self-reward and so on. So that's the approach we took. And we found three strategies from this. One is what we talked about first, pull-in rhythm strategy. There's a dependencies between two animals' pools. And we discovered two types of gaze and pull gaze-related strategy. Basically, we call this one, first one, social attention strategy. So this is basically, one animal is pulling and the other animal is looking at that. So that's the edge here, for example. Another one we found is gaze lead pull strategy. This is when one animal looks at other animal and transition that to pulling behavior. So that's edge looks like this, looking and then pulling their own own lever, for example. So now we have this pull in rhythm strategy, social attention strategy, and gaze lead to pulling strategy that's discoverable in this task. So we wanted to see, are they functional, are they relevant? So we can now starting to look at how these strategies from the model is related to their behavior. So that's what we did. And I'll show you two types of results that speaks to that. One is that these gaze-related strategy were differently used between successful and failed cooperative pools. So which is shown here. If you look at the modulation index that I talked about briefly, comparing successful to failed pool, and you can see that both social attention strategy and gaze lead pool strategy are significantly used for a successful pool compared to failed pool. So that's a good sign. That means there, more of these strategies are used, more successful they are compared to failed ones. And the next one we want to test is if these strategies from the model are really a good, robust strategies, then we should be also able to predict when animals are going to pull based on this strategy. So we made generative model of this and basically calculated prediction of pulling behavior using area under the curve. And let's look at these four conditions. The first is learned strategy, that's including everything. And you can kind of see that's the kind of almost like a default of everything they're doing, success AUC value. If you just look at the pull in rhythm strategy from the model and predict the animal's pull, it's significantly above chance. So they can still do that. So that is a pretty good enough to predict animals pulling, but not as good as learned strategy. And if you just look at the gaze and pull, also it's above chance. So they can still predict, but not as good as learned strategy. But when you combine just the two strategy, pull in rhythm, sorry, just these three strategies, pull in rhythm and gaze and pull strategy, you can actually recover back the whole launch strategy. So basically, even though these are, pull and gaze strategy, there could be many other strategies that one might be not capturing because it might not be robustly used all the time. Even though we just use these two strategies types we pulled out and combine them, they can kind of mimic the full launch strategy, which means that these strategies are used heavily. So now, going back to the advantage of using marmosets compared to, let's say, macaques is because they're small, we can also have more animals, which also means we can look at social relationship better in these animals. And I'll show you some social relationship effects in the next few slides. But first thing that's really noticeable is dominant animal tends to be more active. They're more motivated. So you can see that here, social gaze count is higher for dominant than subordinate. Also, automated lever pull count in that control condition even is higher for dominant. So even though it's hard, they tend to try more in the dominant animal. And this is kind of interesting finding and fits with the literature because females tend to be dominant in this species, and females tend to have more food motivation in this species because they have to raise young. They give birth to twins or triplet every single time. They're all about raising families, so they need extra calories. And dominant animals also used a more strategic social gaze. So going back to this lever gaze over social gaze plot and comparing that to mutual cooperation condition, with respect to automated lever condition, dominant animals are directing attention to that, automatically moving lever more than subordinate animals. Um, and you can look at the mutual cooperative versus self-reward condition. Dominant animal are using social attention strategy and gaze lead pull strategy more. So they're just using social gaze strategy more than subordinate animals, potentially correlated with they're more active, they're more motivated. We can also look at different sex pairings and within kin and non-kin groups. They live in family units, so we can test this. So what's really fun to talk about here is that, they're all about family, and the female tend to be the dominant one in the family group. They don't like to you work together with stranger females. So when you look at this reward count of stranger females, basically zero, almost zero. So they just like sit in the corner. They refuse to do the task, which is really interesting because it's there for them to just get reward, but they just don't want to do it. They just don't want to partake in it. So I think that really speaks to this non-kin competition among females in this species. Whereas if, when you're kin, like daughter and mother pairing, they do pretty well. And male, even the non-kin, they do better than female non-kin. And generally, what's better is female-male pairing. And I think they're just more structured in their dominance too. So they tend to have more roles they play, so they do better than mixed sex pairs tend to do better. So the final part about the behavioral side is I wanted to mention that these animals are not just doing the task in that context, but they're willing to work with one another. So we wanted to test how flexible their strategy is. So we basically tested, across days, partner is changing A, partner B, partner A, B, and so on. And the idea is that, we pick this partner so that the behaving animal strategies can be tested going back and forth. Because we noticed that this animal works one way with the partner A, but the other way with partner B. So here's a basically like before the video, quick summary of that. If you just look at the self to partner pull ratio and of the ginger, is a monkey, when the partner is Dawson versus Kanga, you see that up and down pattern there. So this simple pull ratio is changing, right? And I'll show you the video, and it becomes really clear how ginger works differently with Dawson or Kanga. So here is this is 1.5 speed. Ginger is working with Dawson. Ginger is always pulling twice. One, two. And you can hear chirp, which is a reward sound. Ginger is doing one, two, one, two. The reason why pulling twice is good is it actually increases your cooperation window more than 1 second, right? So it's helping the other guy. And now Ginger, same monkey, is playing with a Kanga, just one time. One, one, because Kanga is better. It's one. Yep. You can kind of see that, depending on the partner, Ginger's behavior is different. Dawson is not that great at this. The Ginger pulls twice to open up the cooperation window. Kanga is very good. So sometimes, you even follow sometimes. So you can see that, the probability to pull is nicely peaking for Kanga, but it has this, shift because you're pulling twice for Dawson. And also social gaze count is higher for Dawson when Dawson is not as good, so they're communicating more than Kanga. So with that, we decided to record what's happening in the brain during this kind of social gaze behavior. How do they make these cooperative decisions? So now we developed this wireless recording system that we can put data logger in. This is basically like a USB stick that can collect neural data from this one is 64 channels, but now we're also developing neural pixel version of the wireless recording. And then we targeted Brodmann area 8B and 9. This basically dorsomedial prefrontal cortex. And we targeted there because that area has, implication in social decision making, also implication for theory of mind. So we're interested in how this region is using information about others to make cooperative decisions. So we had a hypothesis, what mechanisms guide its decisions, and this might be familiar to many of you. So there's like a evidence accumulation framework from perceptual decision making that over time you accumulate evidence. If the evidence is keep accumulating in the right direction for a while, you might cross some boundary and make a decision. In this case, we're thinking in a very analogous way that cooperative decision is when social evidence from the another partner is reaching some threshold so that, okay, I'm going to do it to make a successful cooperation work. And the key signature of this in the neural activity is that, when you look at the neural activity, there's a slope that's corresponding with, evidence accumulation model where drift rate, the slope of this activity is correlated with how much you're drifting, how much you're getting to that goal, basically. So hypothesis we had was this. So hypothesis is that they're doing cooperation, they use gaze a lot. So maybe social evidence accumulation is happening in DMPFC, and the information about the other is coming from social gaze. So social gaze is used to gather information about others' behavior to know, okay, I have enough information that you're going to engage, so I'm going to engage too. That's the idea. Um, Going back to the earlier part, we track this face like this, and their eye movement is about 15 degrees to each. So you can actually, it's pretty small range, so you can actually look at the where they're looking at it pretty accurately with this line coming out. And we have to define what kind of time window we're going to look at. So when you look at the interpol interval between the pool, you see there's like a similar timeline between two animals we tested here, meaning that's when they're kind of engaging timeline. So now to link pool and gaze to this accumulation model, you can think about it this way. So there's a level pool going on, there's social gaze going on. Now, if you make the social gaze into density plot and zooming in one of these, let's say, and you can kind of see that social gaze probability is going up, it always peaks right before the pool. And you can basically now calculate area under the curve to see how much is getting bigger before the pool. And in fact, if you look at the distribution of AUC value, density values, you can see that this is a pooling time. And always the highest density of social gaze happens right beforehand, which means that's when it's consistent with the idea that gives me the enough evidence there, so I can now pool now. And this time is actually a little bit earlier. This is the pool registration time. So start of pooling happens like I don't know 200-300 milliseconds before that. So right after the peak time. And what's tricky and interesting is because there's no structure in this task, there's no trial, right? There's no trial start or anything. Animals doing this continuously without any cue. So we have to think about how to even define trial onset to start thinking about this kind of evidence accumulation model. So we looked at 8 different variables, including looking, angle of the other's gaze, angle to the lever, distance between the two, and so on. And you can kind of see they are very well correlated. So this is like an issue that you face when you're doing naturalistic experiments. A lot of behavior variables correlated. So we have to think about how to decide which of this variable is most informative. So we used a GLM model, and we put in basically all of these variables in our kernel. 80 weighted features went in to discover when they're pooling. And you can see the response time profile is very similar between the two animals, which is a very good thing because response time is super important for evidence accumulation framework. So first, we looked at variable importance, basically looking at the drop in area under the curve across all these 8 variables. And you can see that across two animals, that linear speed of the face, LS, how face is moving, is consistently important for both animals. And we can also test this in a variance inflation factor, how much variance is inflated compared to the other. And you can see that in both animals, linear speed of the face is a good choice, important thing, basically. And you can kind of plot this, more zoomed in way, you can see the linear speed of the phase fluctuates right around the time of pulling. And you can define now, the lowest point of linear speed of phase when the phase is not moving to the time of pulling as a response time. And linear speed of phase is important because, when they pull, they go and drink juice, and then they come back, and that's when the speed is lowest, and then start moving towards the lever again. So it's like a functionally very intuitive in many ways. Um, and then when you think about, drift diffusion model, like a DDM model, there's a drift rate going up, and we're thinking the partner's behavior is giving this evidence, right? So we have to also think about what kind of behaviors of the partner we're going to use as, evidence. So learned from the automated lever condition, we think partner's whole body behavior is very important here, not just the hand, for example. So we decided to do PCA of all these variables of partner, those 8 variables, and found that, first PC1 explains, huge variance between the both animals. So we decided to use PC1 as a holistic summary of partner's action. So now we have self-paced trial, self-behavior, and partner's action behavior that animal might be using. We can now test how these variables are actually used to guide this drift rate. So we tested many models, and this is the best fitting model. So basically, drift rate V is modeled by linear speed of action actor and the noise of that. Added partners PC1 and partners PC1's noise, which is uncertainty of that, right? And what's interesting here is the best fitting model actually was best fitting only when we included these variables when actors looking at the other monkey, right? So if actors already not looking at the other animal, adding this all sorts of partner behavior PC1 doesn't do much. However, when actors looking at the other animal, adding this PC1 of partner becomes really important and thus led to the best fitting model. That's what's shown here. Importantly, what we find here to be critical is this beta 4, which is basically the noise term of PC1 partner. So if you're thinking about like gathering information, social evidence information from another, uncertainty and the noise level of the partner one's behavior is contributing that a lot. Higher the noise, lower the evidence, lower the noise, faster the drift rate. Basically, that's the idea. And you can see that there's a nice negative correlation for both animals, indicating that, drift rate is slower when partner's behavior is noisier. And you can visualize this here. If you pull up peaceful on a partner, and variability partner and the gaze of actor are, actually happening at around the same time. And this is a pull time. So this kind of really makes sense here. So, DDM modeling suggests the social gaze mediated, this holistic action of partner is important. And especially this noise term is really important, that guiding the drift rate. So now with that, we can now look at do neurons in the DMP actually track these variables? So we can align at the time of pull time and look at the reaction time, response time, and to see if they're consistent. We can also align at the trial start to look at the evidence amount to be important or not. So here's the 2 examples cells here. You can see this is time of pull. Aligning backwards, you can see highest response time is lowest drift rate. Low reaction time, response time is highest drift rate. And across all cells, you can see the very strong negative correlation across these 2 variables. So consistent with the drift diffusion model in the RT domain. You can also align that to trial start. Now we can look at high social evidence. This is low, the noise term, and low social evidence, that's high noise term. Evidence faster the rate of evidence accumulation. And here's a population summary of this. You can first see that neurons that track the slope in general are else than the failed part. And you can see they're both animals. And what's interesting, um, all the neurons actually tracking the evidence are subset of that, 100% basically, which means that neurons that are doing this are also neurons doing this, which kind of puts them together in the same kind of functional space that social evidence accumulation tend to be encoded in these neurons. You can also make a neural drift diffusion model, basically plugging in neural activity directly to the model to see do they track rate of evidence accumulation V or starting point, the prior kind of point Z. And here's data from there. If you look at the V equals slope firing rate, you can see that a lot of neurons encode that. So that's, even the neurons model is working. And also, many neurons also encode this prior point. So there's like prior information as they're behaving, that information also reflected. Here again, the neurons that are encoding this drift rate directly, right? Encompasses all the neuron that encode this, more or less all the neuron that encode this starting point. So there's also functional overlap in this sense. So finally, because we recorded multiple neurons at the same time, we're very curious if there's some kind of neurodynamics at the population level that's corresponding with the social evidence accumulation. So in that case, we did PC1, PCA, and then looked at trajectory of neuron firing rates over time from start of the trial that we defined self-paced trial start and the pooling action, and looked at how they kind of evolve over time. And in fact, specifically, we looked at the past variability across trials with a assumption or hypothesis that if the evidence is high, you don't have to vary that much. You can do trajectory that you're familiar with in the neural space. But if the social evidence is noisy, then there will be a lot more variability in evolving time to that pooling time. Um, and yeah, that's what we looked at. And that's what you see actually. In both animals, the past variability term is negatively correlated with social evidence. So, variability is more variable when the social evidence is low. And interestingly, that relationship is only there when animals are successfully pulling. So when they're doing failed pull, this variability information linked to social evidence is not present. So they're just not doing that similar calculation. But when they're successful, then the noise of this past variability in the trajectory space is now correlated with social evidence. You can look at this in another way. Look at the social evidence low to high over normalized time point. You can see that, the variability strength, which is color coded here, is much stronger in the beginning than in the end, which is consistent with idea in the beginning that you're more noisy, more uncertain there. But then as animals are moving in and getting ready to go, your uncertainty is getting reduced. So the noise variability is more stable. So that's another facet of this trajectory kind of analysis. Okay. So with that, I want to give a summary of of the results. So cooperative strategies in marmosets seem to be very flexible, and it's really tuned to a social partner. They're not just coming to the task and this is my context, this is what I do kind of situation. They're like, let's work together to solve this as well, and that's what's reflected in the partner-dependent change in their behavior strategy. It seems that neurons in the DMPFC implement this gaze-dependent social evidence accumulation, where social gaze is used to accumulate this partner's PC1 variable to the point that, when you reduce uncertainty enough, now that is basically functionally same as crossing the boundary for me to reach out and pull. So one interesting for us is that, all the evidence accumulation kind of framework has been done more in a structured setting. You have trial start, you have some kind of task time period and so on, which makes sense to model that really easily. But we found that if you accurately define the behavior carefully in a robust way, you can still apply this kind of canonical model, even though when things look like out of control in naturalist behavior. So you could actually, use what we know in the brain, canonical framework can also be used in this kind of naturalist setting. Um, and finally, it seems that this kind of flexible, diverse strategies in the MPFC, kind of informs how this kind of cooperative interaction that take into account others' behavior and maybe even intention comes into play in the primate brain. So with that, I want to thank everyone who participated in this work. This work was all by Wei-Kang Shi and Olivia Meisner in the lab. And I thank my two collaborators. We work on these projects together and funding sources. And thank you for listening to this talk.
Thank you so much, Steve, for this highly interesting talk. We have about 5 minutes for a couple of questions, point for discussion. Please raise your hand and I call you, or you can also directly type into the chat if you like.
There's someone?
Say in.
Hello, can you hear me well?
Yeah.
Yes.
Okay, thank you. Thank you so much for your talk. As a primatologist, I was wondering about the point that you mentioned about the noise in your DDM, this model, because in wild primates, there are also the noise coming, well, generated by the third party intervention. And I was not sure whether you meant this noise only about the second party or also including the third party, and whether your model can also reflect this.
That's a great question. So in our context, all the noise comes from the behavior of the partner because everything else is completely fixed. However, what you're saying totally makes sense. If there's a lot other things going on, that will add to the noise as well. Um, you could capture that with another set of terms that are like, other noise or environmental noise. But what I might predict is as long as the task success is dyadic kind of thing, this drift rate will probably be still differentiate based on the partner's noise level. But maybe the whole thing will be kind of generally slower if environment is very noisy. You know what I mean? So more distracting in general. Yeah. Thank you.
Any more questions?
So since we have time, I can also add that we also did altruistic version of this task. If I can see that slide. Oops. Um, Oh, I don't see it here. But basically what we did is like, would they do this task in a completely altruistic setting? So we basically have animal pool without any reward, but only delivering reward to the other. And then after like 30 seconds, we take turns back and forth, back and forth. And in the preliminary data in one pair, they have a nice correlation of pooling count across blocks. So they are also being able to do more like a reciprocal altruism type way of doing this lever pulling. Um, yeah, and another thing to mention is like when we're thinking about this design, the you know abstraction really matters, it seems. So like humans, we can do tasks that are a lot more abstract because we are really good at that abstraction. But animals are sometimes good, sometimes not good. So, if you're designing like a certain task that requires a little bit more abstraction to be successful, we try to reduce that as much as possible. And I think marmoset model is easier in marmoset model to reduce that abstraction compared to some other model systems. So that's another kind of interesting thing that, maybe monkeys and humans can do many types of complex social cognitive stuff. But maybe the bottleneck sometimes that people fail to find certain cognition in certain animals could be that, you might have to like tailor your abstraction to that species. If you don't tailor it, maybe you're not even engaging the right system or the circuit for that types of behavior.
Maybe a very basic question. I guess you get this a lot. From a comparative standpoint, what would you say is your main take-home message or takeaway from this, what we can learn for humans?
Yeah, yeah. I think the marmosets are strong model for certain aspects of human social cognition that is really kind of in-group oriented and the family oriented kind of social cognition because they are all about family. They're like really strongly tied to this kinship and non-kin. Um, uh, on the other hand, if you're more interested in more like strong dominance and competition and resource fighting, then macaque is a lot better model for human social cognition. So these animals are so socially tolerant, they're totally fine if a subordinate steals their food. They don't care sometimes. They're like, okay, you can have it, I'm fine, I'm cool, whereas macaques go crazy, right? So I think it models certain aspects of like a really kind and friendly, family loving side of human social cognition really well. So I think it's good to think about even within the primate species, there might be like different model systems that are great for modeling certain aspects of human social cognition. In terms of brain areas, they're like really similar, right? So I think what we learn is it will be like really surprising what you learn from the primate brain is not there in the human brain.
Okay, thank you very much. And yeah, thanks again so much for this interesting presentation. I'm afraid we ran out of time. We'll keep you posted about the next talk in our series. And yeah, have a nice evening, morning, wherever you are. See you around.
Thank
Bye.
you so much. Looking forward to learning about other talks.
Bye.
Bye.
Thanks,
Bye.
Steve.
Thanks.
Thank you
Bye.
so
Good to
Bye.
much.
see you.
What a
Good
cool
to see you,
talk.
Kendra. Thank you. Bye.